This is an OCR A-level biology video on mitosis. I might put meiosis in this video as well if I don't spend too long, and definitely in a separate video I'm going to look at differentiation of cells in tissues, organs, and just looking at stem cells. That'll be in a different video. If you've not seen the cell cycle, I advise you watch that before you watch this. But anyway, let's just get into mitosis. So mitosis is needed for the growth of multicellular organisms, for repairing damaged tissues. It is also a method of asexual reproduction. So it's got these 4 stages: PMAT, prophase, metaphase, anaphase and telophase. It's really one continuous process, but we sort of put it into these stages. Okay, interphase becomes before mitosis in the cell cycle, and interphase is on my previous video. So we use it for growth, tissue repair, asexual reproduction, etc. So first of all, let's look at a cell that comes out of interphase. So when the cell is finished interphase, it looks like this. 2 lots of DNA, all the DNA has been replicated in 2 centrioles or centrosomes containing centrioles. The centrioles or centrosomes migrate to separate poles, one this end, one that end. This is still the same cell, but I've just drawn it like this now to make it easier. This is the first stage, so the nuclear envelope breaks down and we see our chromosomes coil and condense into the forms you probably recognise chromosomes as. So these sort of X-shaped structures with a yellow dot in the middle representing our centromere. Let's just pause it there a second. So this first stage is called prophase. So the nuclear envelope, the membrane around the nucleus, breaks down. So then we get our chromosomes coil and condense, and our centrioles or centrosomes containing centrioles migrate to either poles, and they start to form a network of fibres or microtubules, and we call this network the spindle, and that is in grey. So you've just got to remember centrioles at either end of the pole. So if you think about the Earth, you've got the North Pole and the South Pole. They're the opposite ends of the Earth. It's the same in cells, opposite ends of the cells, so we call them poles. One centriole here, and they both form these microtubules, and it's called a spindle. These microtubules will then attach to our centromere, which is in the centre of our chromosomes. So that's when the next stage starts. So metaphase, PMAT, remember. So we've just seen prophase, now metaphase. So I draw it on all again. Our spindle now, which is these grey fibres, attach to our chromosomes, and I don't know why it's drawing it out like this. It's just something that the program does. Let's speed it up a bit just to get rid of that. So we see this spindle formation, okay? So they attach to the centromeres of the chromosomes, which are the yellow dots, and they also attach to each other. So this spindle is formed. We see them line up in the middle here. So here I've got 2 chromosomes, but in an actual one, of course, there's going to be 23 pairs. So 23 are going to line up in the centre of the cell, and they will attach to these centrioles by spindles. So the chromosomes with 2 chromatids, so let's remember from the last video, that's a chromatid, sister chromatids, together they make a chromosome, but one on their own without this centrosome makes is also called a chromosome, which is quite peculiar, but you can watch the other video if you don't really understand that. They line up on the middle of the cell and they become attached to the spindle by the centromere. So in the previous video I spoke to you about the metaphase checkpoint, and I didn't really tell you what happened. So the metaphase checkpoint is basically checking that all these chromosomes are attached to these centrioles, so making sure these microtubules attach to the centromere and making sure that they're all in the centre and lined up. So once that they've passed that checkpoint, once everything's attached and everything's in the right place, then we can move to anaphase. So that was metaphase. Meta, middle, lining up on the equator of the cell, microfibrils attaching to the centromeres. Okay, so now let's have a look at anaphase. So we're seeing these microtubules pull our chromatids apart. So here we've got a chromosome. I might actually draw this on here. So that is a chromosome. They get pulled apart. So one gets pulled that way, one gets pulled the other way. Okay, and these individual things are now chromatids, and they formed this chromosome before. So what happens in anaphase, where they were joined up, they are pulled apart, okay, by the centromere, and the centromere is pulled first towards the centriole, causing this V shape, and the V's pointing outwards. So the centromere is pulled towards the centriole, and we're seeing these chromosomes split apart into their chromatids. And what was the centromere we now call the kinetochore. I'm not sure if you have to know that in this specification, but because the centromere is split in two, it's still attached to something, but we then call it the kinetochore. However, you can probably ignore that fact. So that is anaphase, literally just pulling them apart to the separate poles, centromere first. Then we have our final phase, which is telophase. And telophase is when the chromatids reach the opposite poles, and they uncoil, become long and thin again, and they are now called chromosomes again. Okay, so where they were chromatids, once they are in this nuclear envelope again, they then become chromosomes, which I understand is painful to try and learn, but it's just the way it is. That you now call them chromosomes. The only time you call them chromatids is when they're being pulled apart. That's when they're chromatids. Chromosomes is when they're in this nuclear envelope. So our chromosomes are at separate ends of the cell. We've got the nuclear envelope reform in telophase. Our centrioles are at opposite ends, and as you can see, we're getting this thing here called a cleavage furrow, which is the start of cytokinesis, which is not part of mitosis. So mitosis is the division of the nucleus, nuclear division. Cytokinesis is not part of mitosis. It's part of the M phase of cell cycle. Is not part of mitosis. It is the splitting of cells, so the splitting of the cell membrane into two new cells. So mitosis is the splitting of a nucleus, and cytokinesis is the splitting of two cells. Let's just run through how cytokinesis works. So we get this cleavage furrow, and that will keep extending here. I don't really show it in the animation, but then we get two new cells. And let's just remember that we've now got two diploid cells produced, and they're both genetically identical daughter cells. So this one cell has produced two genetically identical daughter cells. And you might be thinking, well, it doesn't make sense. How can we get 23? Because you're saying this cell's a diploid. 23 into 23 and 23, it doesn't add up, does it? But let's remember, at the end of interphase, we'd doubled the amount of nuclear material, so we've doubled the amount of chromosomes. So really we go in with 46 and we end up splitting that 46 into 23 and 23 pairs of chromosomes. But this is still called a diploid cell even though all the DNA is replicated. Okay, so a diploid produces two new genetically identical diploid daughter cells. And that is pretty much everything. I'm going to split this video into another two, so the next video I will do meiosis on its own, and then after that we will look at differentiation of cells. Okay, all the best, see you next time.